The overall goal of this live cell Főrster resonance energy transfer, or FRET-based assay, is to assess G protein-selective G protein-coupled receptor, or GPCR, conformations under different agonist conditions.
This method can help answer key questions in the GPCR field about GPCR interactions with different effectors and the effect of different drugs on receptor conformation. The main advantage of this technique is that the sensors are modular and can be easily re-engineered for different GPCR and G-alpha peptide combinations, including the full-length G-alpha subunit.
Though this method can provide insight into G protein selection in response to different ligands, it may also be applied to explore the physiological consequence of differential G protein activation.
Before beginning the procedure, culture the cells of interest in complete medium at 37 degrees Celsius in a humidified atmosphere at 5% carbon dioxide until the cultures reach confluency. Next, detach the cells with trypsin and passage the cultures at 8 × 10⁵ cells per well in 2 millimeters of medium into tissue culture treated 6-well dishes for 16 to 20 hours of culture. After 16 to 20 hours, prepare 1 transfection reaction per well when the cells have adhered to the plates in a biological safety cabinet, and allow the reagent mixtures to equilibrate for 15 to 30 minutes. At the end of the incubation, add 1 reaction to each cell culture dropwise across the well, gently shaking the plate to ensure a thorough distribution of the reagent throughout the